So I'm going to talk today about something that's very close to my heart, and after all these wonderful hors d'oeuvres that we've eaten, hopefully very close to your hearts, and that's our GI tract. And specifically, I'm going to be talking about the microbes, the bacteria, the germs that are present in the GI tract. The name of our study is the St. Louis Gut Neonatal Microbiome Initiative, otherwise known as the Twin Study, for reasons that you'll see in a little bit. And our collaborators include Dr. Philip Tarr, who's here in our audience. Dr. Tarr, who's the Director of Pediatric Gastroenterology and Nutrition here at St. Louis Children's Hospital, Division Director, and Dr. Jeffrey Gordon, who's the Director, Dr. Gautam Dantes, who's a scientist at the Center for Genome Sciences and Systems Biology. And we also collaborate with many of our community physicians, as you'll see. So I'm here to tell you that, as Dr. Schwartz alluded to, you are not alone. And in fact, as a fact check for him, there are in fact 10 times as many microbes present in your intestines right now than are present on the entire human body. And by far and away, the gut is the major reservoir of those bacteria. There's over 100 billion bacteria present in your intestines right now. So let's hope they don't have a revolt. That's more than all the people on the earth. The microbiome is the combination of those bacteria and all the genes, the DNA, the genetic material that's present in those bacteria. So it changes the emphasis a little bit from just looking and identifying what bacteria are there to also being able to identify what is it that those bacteria are doing. What are the genes that are present in those bacteria, and what functions do they give to that bacteria? We've known for really a long time now that bacteria can do a number of things that our bodies can't. And we're in a symbiotic relationship with that bacteria. So they can make vitamins that we can't make, they can break down certain nutrients that we can't break down. But what we're really just starting to understand is that those bacteria are intimately related to our body's function, not just in our gut, but really systemically. And that's what I'm going to be talking a little bit more about today. So why study the infant? What's so big deal about the baby GI tract? Well, when you're born, you're actually born with a sterile gut. You don't have any bacteria present in your GI tract. And over the first weeks to months of life, you start to infill your GI tract with bacteria. So if you're a baby, what are you doing? You're feeding, you're sucking on your hands, you're eating a little bit of dirt, you're chewing on the dog's tail. So the gut is really a portal of entry for those bacteria. And some of those bacteria can be harmful, like the listeria that we've heard about in our cantaloupe outbreak. And some of those bacteria can be what we call good bacteria, commensal bacteria. And somehow your body has to figure out which is the harmful bacteria and which is the not harmful bacteria. And the way it does that is through the immune system that's present in our GI tract. It's really the guardian of that portal. If it identifies something as harmful, it's going to trigger an inflammatory response, and it's going to get rid of that harmful bacteria. But if it identifies something as not harmful, it's going to tolerate it. It's going to allow it to be there. It's not going to have an inflammatory response. And what we now know, really, just in about the last 5 to 7 years, is that it's the pattern of good bacteria that are present that helps keep this balance between inflammation and no inflammation in our immune system. And if you disrupt that balance and that pattern of good bacteria, or if you never develop that kind of pattern at all, then that will lead to a propensity for too much inflammation. And if it's too much inflammation, It can result in local kind of problems, local diseases in the GI tract, like inflammatory bowel disease, or it can potentially spread systemically with too much inflammation in your system and promote the development of autoimmune disorders like allergy, asthma, and even diabetes. So the second systemic effect that I want to talk about is this idea of energy harvest. So I have a 17-year-old daughter who's a skinny little thing, and if she eats 1,000 calories of something, she may harvest only 500 of those calories. But if I eat those same 1,000 calories, I know I'm going to harvest every single calorie that's in there. And part of that difference is related to differences in the microbial communities that are present. Now, sometime in our evolution, there was probably an advantage to having a microbial community that was able to harvest every piece of calorie and convert it into fat when we were hunters and gatherers. But I was just watching my husband the other day. The only hunting and gathering that he's doing is playing with these angry birds present on his iPod video, and there's not a lot of energy expenditure with that. And so that evolutionary advantage is now a potential disadvantage in terms of obesity. Now clearly, the microbiome, and I'm not trying to tell you that it's all the microbiome's fault, but by knowing that there's this difference in the ability to harvest energy based on the microbial community, that has the potential for intervention both in terms of overnutrition and obesity, as well as undernutrition and malnutrition. And then finally, the third systemic effect is this idea of antibiotic resistance. So what's antibiotic resistance? Well, the bacteria no longer respond to the antibiotics we use to try and get rid of them. And the reason that they develop antibiotic resistance is that they acquire little pieces of DNA that confer that protection to them. And the more antibiotics that you're exposed to, the more pressure there is on these bacteria to acquire that genetic material. And we just heard that the biggest reservoir of microbes in the human system is through the GI tract. And how do we take most of our antibiotics? Usually we're taking it orally. So could there be a relationship between the development of antibiotic resistance over time? And that's one of the things that one of our collaborators is looking at with us, with the gut microbial community as a reservoir for those antibiotic resistance genes. So, as you can see, all of these patterns really begin early in development, and that's why it's so important to look at the infant microbiome. Oops. Okay. So why study the microbiome now? Well, we just reviewed that it's recently been recognized that it has importance far beyond the GI tract. But the second reason is that within the last 10 years, and really even within the last 5 years, we've developed much better tools to look at those microbial communities. So we used to have to grow the bacteria up. And in stool samples, for example, over 50% of the bacteria never even grew. So in fact, we didn't really even know what it was that was in there. We now have technology that doesn't depend on having those bacteria grow. And it does it faster and increasingly cheaper. So you start with a microbial community, and it used to be that you put them on something like these culture plates that were at our poster. But now, instead, we extract the DNA, and then that DNA is sequenced. So we look for the composition of the genetic elements that make up that bacteria. And then we use computer-based technology to help us identify what bacteria are present. And not only what bacteria are present, but what are the genes that those bacteria have, and what are the functions that those genes can do.